Ein Glas Wasser? Ja, bringe ich Ihnen. Frau Gutlerlein. Hat er schon? Yes. So, okay. Yeah. So, welcome back. Hope you have good energy for the second part of our symposium. There were some questions on the schedule of the week. You all have this printed program, and on page 9 you have the actual schedule of the week. So, you can find it in your program on page 9. With these words, I would like to hand over for Max for introduction.
Welcome back. Our next speaker is Wim Rijbroek, if I spell it right. He's lab manager at the Technology and Food Science unit within the Flandern Research Institute for Agriculture, Fisheries and Food. And he is in many groups at IDF, and he is also chairman of Group 16, for example, or chairman of Group A18. And he will talk about antibiotic testing in Germany and around the world. Welcome.
And also about the new standard ISO TS 23758 IDF RM 251. Recently published. Great.
That's what
okay. Thank you for the introduction. I'm happy to be here and to meet you again in person after more than 2 years of isolation. So I will mostly talk about that publication about the new ISO technical specification. If it's, do I have to, for the next slide, the green button? But no. Okay, as introduction, I would like to tell that compared to other food matrices, milk is quite a clean business, especially when you talk about the presence of antimicrobials. So in food of animal origin, there is always a possibility of presence of residues of antimicrobials, and you can see these are the official data published in the latest EFSA report. It's about the national monitoring plans. And if you compare milk, and this is based on quite a reasonable number of samples, you can see that the occurrence of non-compliant results is quite low compared to other food business matrices. So we have overall European figure of 0.07% of non-compliant results. And this is just like with speed on the motorway. If there is no monitoring, everybody will drive too fast. It's just the same here regarding presence of residues of antimicrobials. And this is due to the frequent testing. If we look to the whole system, so there is many spots during the flow from milk from the dairy farm up to the consumption milk in the supermarket. There is quite often sampling and quite often monitoring for presence of antimicrobials. It's starting already at the dairy farm, where the dairy farmer himself can check the milk of a treated cow before he let run the milk into the milk tank. He can also check presence of antimicrobials in the milk of the milk silo. Further, you have the official sampling, called the sampling of ex-farm milk. When the tanker is arriving at the farm, an official sample is taken, and in some countries, every sample is analyzed on presence, like here in Germany, every 4 to 5 samples in a month of these ex-farm milk samples are checked on presence of antimicrobials. Arriving at the dairy company, there is the obligation to check for the presence of beta-lactam compounds, so to the presence of penicillins and cephalosporins with a rapid test. And many dairies also check the big silo, the content of the big silo before going into production, mostly with a broad-spectrum microbiological test. And finally, you have the control by authorities, the check of consumption products in the supermarket or on the way part of the national monitoring plans. So in Germany, if we just look to one milk control station, if we look to the data for Milchprüfung Bayern, these are the data in the year report from last year, 21. So they are checking around 25,000 farmers in general, in average, and they're checking 4 to 5, they are running 4 to 5 antimicrobial tests per farm per month. And this is resulting in a huge number of tests. So 115,000 tests per month, just for one single lab already, 1.3 million, above 1.3 million antimicrobial tests per year. And they are using microbial inhibitor test, the BRT high sense, and their positive percentage of samples, it's 0.085% of positive results, resulting in a penalization, a price deduction of 0.03 euro per kilogram of milk delivered and calculated over the whole month. Yeah?
This is the number of farms penalized.
The number of farms penalized. So that's a misinterpretation of that last data. Okay. So for testing, there are a lot of tests available. You can split them up in between screening tests and confirmatory tests. And the screening tests you can further split up between microbial inhibitor tests based like on disk assays, and you also have agar diffusion tests. A lot of rapid tests are daily coming on the market, mostly based on lateral flow devices. There are also labs still using ELISA assays. So there is a whole market of rapid tests. And monthly new tests, if you look in the hall besides, you see always every month new tests arrive on the market. They are faster, they are easier, they are more sensitive, or they are detecting a broader spectrum of antimicrobials. But the problems still remain for the user. What is the intrinsic quality of the test? If you want to buy and use a test, what is the intrinsic quality of your test? Is your test fit for purpose? Because most of the tests are developed for raw cow's milk, but some users also want to test goat's milk, ewe's milk, or they also want to test heat-treated milk. And they still remain with the question, is this test suitable for that aim? And therefore, there is only one solution, and that is validation. And validation is determining of if the test is fitting for purpose. And so it's an important element of quality control. And validation helps to provide assurance that the measurement will be reliable, that your result is reliable. And even in some fields, it's an obligatory part. So if you want to go for accreditation, you must first of all use validated methods. So you have to validate for each new developed method. If you revise a method, you have to redo some validation work. And also if you transfer to a new lab, new validation work, it's more a verification instead of validation, if you want to use that wording, if you transfer to a new laboratory. So there are many validation parameters popping up for an analytical test. So you have accuracy, you have precision, you have specificity, selectivity, you have linearity, you have limits of detection, limits of quantification, you have your range. So in European legislation, we have a Commission implementing regulation, the 808 from 2021, indicating which parameters a lab should validate. And they make discrimination between screening tests and confirmatory tests. Most of the screening tests used for the determination of antimicrobial residues are qualitative screening methods. So the lab should validate the parameters CC-beta, or detection capability, should validate selectivity, specificity, and robustness. And you in this legislation also the parameter stability, but stability, a lab could refer to data published in publications, so data available in literature. So we have guidelines, so we know which parameters we need to validate. So the legislation is giving guidelines. Also, the community reference laboratories published guidelines in 2010 for the validation of screening methods for residues of veterinary medicines, both for initial and transfer validation, but however, precision is missing. And there was a gap in knowledge how to validate, and there's also a gap in harmonization. And this gap was filled up within this organization of IDF ISO. There was an action team formed, and this resulted in the publication last year of an ISO IDF technical specification, and this was with as team leaders Valérie Godet and myself. So these are the guidelines for the validation of qualitative screening methods for the detection of residues of veterinary drugs in milk and milk products. And now I will go a little bit further in detail in this technical specification. Very important, this document is also taking over the idea of an initial validation who is applied to a newly developed method, and where one single lab is fully validating the method. This lab is called the originator laboratory, and that lab is demonstrating that the method is fitting for purpose. If any other commercial or other smaller lab wants to take over the data, they just have to do a verification. And we call this an abridged or a transfer validation. So this is a shortened validation procedure applied in the receptor laboratory on condition that this laboratory has access to the same method, applying the same method, and have access to the full original validation document. So let's start first with the big work. The big work is the initial validation done by the originator lab. So the first parameter to be checked is the detection capability, or CC-beta. This is the smallest content of the analyte that can be detected, identified and or quantified in a sample with an error probability of beta. And the beta is defined in European legislation. For screening tests, this is the beta error, this is the false compliant rate, and this should be lower than maximum 5%. So, in general, the detection capability should be defined and determined in the specific matrix, and also milk from the specific animal species for which the test was developed, which in most cases will be raw cow's milk. So you can only validate for raw cow's milk, but if you want to apply the method also in milk of other animal species, this should also be involved in the validation work. So the compounds involved in the study is depending, and there are different choices. If you want really a full broad method, all compounds present on the MRL, for in the European situation, on the MRL list should be included. Or you can limit and you can say, I'm only going, typically for my country, I only validate the compounds of all the brands who have a registration in my country. Or you can still further limit and you say, I only look to the compounds who are frequently used and applied to milking cows in my country. So this is a little bit depending on the test. If you are validating a microbial inhibitor test, there is a very long list of compounds involved. Instead, if you're validating a group-specific or even a substance-specific test, this number of compounds will be more limited. The concentrations you want to test to know from which concentration on I have a 95% detection. You can work in very small increment steps so that in between two different concentrations you include in your validation study, this can be very limited. So normally what is proposed here is if you are testing a concentration in between 1 and 10 ppb, you're working in an increment step of 1 ppb. If you work in a range in between 10 and 20 ppb, you use an increment step of 2 ppb and so further on. It's important to note if you are far away from the regulatory limit, from the MRL in most cases, and if you're away below half MRL, you can double the increment step. Even so, in the situation you're testing a concentration above the regulatory limit, above the MRL. In case you have to test a large number of substances, you can make the option and to choose for bigger increment steps. And you can work in increment step, let's say, at MRL, at 3/4 MRL, half MRL, 1/4 MRL, and at 1/10 of MRL. So this is the approach that is mostly followed if you're validating a microbial inhibitor test. The number of replicates, this is coming from legislation. So if you're at half MRL or below, 20 replicates is sufficient. If you're in between half MRL and 90% of MRL, you need to run 40 replicates. And if you're close to MRL, 90% of the MRL to the MRL value, you have to run 60 replicates. That's quite an enormous work. Once you're above MRL, again, you can limit to 20 replicates. It's important to notice that in case you want to have validation, not only for cow's milk, but also for other animal species, you can, in the determination of the CC beta, directly include the other animal species milk into your validation study. And if you have to run 60 replicates, for example, you can split up and say, I run 20 samples with cow's milk, I run 20 samples with ewe's milk, and 20 samples with goat's milk. And at the end, your validation is valid for all three types of milk of 3 animal species. The same if you want to include heat-treated milk samples or other types of matrices, this is also possible to include it from the beginning in the determination of your CC beta value. So you're looking for the lowest concentration giving you 95% of a positive outcome. That means that you must have at least 19 upon 20 replicates positive. If you're running 20 replicates, 38. If you're running 40 replicates, or at least 57 positive results when running 60 replicates. So this is all done in the matrix itself. You're using blank milk. This is milk coming from at least 4 different animals. You must be sure there is no background of presence of residues of veterinary drugs. So therefore we use milk of animals where we know they have not been treated with veterinary drugs in the last 2 months. These animals must be in good health, in mid-lactation, delivering milk with a normal to low number of bacteria and somatic cells. So if you're running 20 replicates, you need to apply at least 4 different milks in your study so that you have some variability, by preference that even the 4 different milks are coming from different milk producers. And this is the outcome. I give as an example. So based on study of initial validation study, here you see the results of the CC beta values for the rapid tests who are passing the criteria set by the Belgian Federal Agency for the Food Safety. So we have acceptance criteria. These criteria here are for the checkup of incoming milk at the dairies, where the rapid test must be able to detect all beta-lactam substances at MRL. Only one can be missed, but should be detected at least at 5 times MRL. And these are the new criteria who coming in application from the 1st of July next year on. You see there are only 2 tests fulfilling and detecting all the beta-lactams who are present in brand names in Belgium at MRL. The others are able to detect all except one, but still detecting at 5 times the MRL. So I'm passing to the second parameter, the second validation parameter, that is test selectivity specificity. And this testing is designed to identify which substances who are not previously identified are detected by the test, so who are possibly interfering. And therefore, we have to include related substances and also representative substances of other antibiotic families in case we are testing group-specific tests. So we dope at high concentration, 100 times the MRL, and we test and duplicate to see if there is any interference. If any interference is observed, these compounds will be added to the list to determine the CC beta value. That we also want to know from which level on these compounds are also detected with a 95% of detection. A very well-known interference for the beta-lactam rapid tests is interference by clavulanic acids. It's a beta-lactamase inhibitor, but also containing the beta-lactam ring. So most of the group-specific beta-lactam tests are also giving positive results for presence of clavulanic acid. We also determine the rate of false positive results by running a high number of blank samples. So in the standard is standing 300 blank raw milk samples and 300 blank tanker milk samples. And if any positive outcome is observed, it has first to be confirmed there is no presence of antibiotic residues. So then the percentage of false positive results can be calculated. The last parameter is test robustness. Test robustness is designed to identify the impact of small changes in the test protocol or to identify the range, the extreme ends of samples in which the test will operate. So, and will still give correct results. And robustness testing includes different parameters. Could be testing of the influence of the test protocol, influence of the impact of the milk quality and the milk composition, testing of the impact of the type of milk and animal species, testing of influence of the batch of the reagents, the production of reagents, and the age of reagents, and finally also testing the stability of the readers and the reagents. If we look to the first group of parameters, and that is the influence of the test protocol, so any changes will be made to incubation temperature, incubation time, delay of reading, setup time, volume of milk, temperature of milk, and the small changes in the protocol are introduced, and the impact is checked, is verified by comparing the results when using the protocol as given in the test kit leaflet. For this situation, we have to test at least 4 blank milk samples and 4 positive samples doped by preference at CC beta or slightly above CC beta. Second group of robustness testing is checking the impact of the milk quality and the milk composition. So normal milk, milk with normal composition, good quality, and from cows in mid-lactation, these results are compared for milk with a high somatic cell count, milk with a high and a low protein or fat content, milk with a low and a high pH, and milk coming from cows in early or late lactation to see the impact if the milk composition or quality is different from the normal situation. And for this testing here, this is performed on 10 samples, 10 blank samples, and 10 doped samples. Also, the testing of milk type and animal species, this is optional. If the test is never aimed to be used for milk different from cow's milk and only testing raw milk, this validation should not be performed. Here we compare the results we obtained for UHT milk, for sterilized milk, thawed milk, reconstituted milk, and milk from other animal species. We compare with the results for milk, raw cow's milk, with normal composition and good quality. And this again is performed on 10 samples, blank samples, and 10 doped samples, doped close at CC beta or close to CC beta. Then the influence of the reagents, so different batches. In the technical specification is specified if you include in your detection capability testing 3 different lots of reagents, you don't need to do extra validation work regarding this parameter. Otherwise, you have to test extra samples using different production lots of reagents. We're also testing the impact of the age of reagents by testing reagents shortly after production and shortly before expiry date. And the stability is daily followed with blank and positive standards. The test and reader repeatability can be also calculated. And it's also advice of participation in the initial validation procedure. The advice to participate, to recommend to participate in a national or international proficiency testing in a ring trial for the detection of antimicrobials in milk. So this was the initial validation. This is a big work for some microbial tests. A lab can be really full-time working for 5-6 months on such a full validation study. And as everybody is understanding, this is not applicable and not necessary that every lab is repeating the work. So if a commercial lab or a smaller lab or another lab want to use the same method, they can take advantage of the initial work done by the originator lab. So the receptor lab has to verify and to demonstrate that they are able to produce the same CC beta values on the scope of samples they are using they are testing in their lab. So on condition that they are using the same test material, the same equipment, they're using the same screening target concentrations and the same cutoff levels, so that they have access to the full report. Once they have proven they can generate in their lab the same CC beta values, they can take over other parts of the original validation study. So this is a significantly reduced version of the validation. So the parameter detection capability, they can limit the work. They only need to verify for one compound for each family of compounds. And by preference, they choose for the most difficult compound to be determined, the one with a CC beta who is closest to the regulatory limit, to the MRL value. So they only have to go for one for each family of antibiotic family. So they only have to validate for one penicillin and one cephalosporin in case they want to take over the validation report for a beta-lactam test. And they only have to run 20 replicates. So they have to only to run one compound for each family, one concentration, that's the CC beta concentration, and only 20 replicates. Once they have proven they reproduce the same CC beta, they also obtain 95% detection in their lab. They can take over all the other CC beta values for the other compounds of the same families. So for test specificity and test robustness, they have to do nothing at all because they are using the same reagents. So they can just take over the data from the original validation report. So they can also calculate their in-house reader and test repeatability, and it's also recommended for transfer labs that they also participate not only once, but that they also frequently participate in an international proficiency testing to prove that they still continue to produce correct results. So I'm coming to my conclusions. The publication of the ISO IDF document, this technical specification, will contribute to harmonization of validation studies, and everybody can agree that the volume of work that needs to be done by a receptor lab is very limited, and that they take the advantage of the huge work performed in the originator lab. I still have two recommendations. Before you start validation work, plan carefully your validation study, because this can limit the quantity of work you need to perform and also limit the costs. And also keep all your data, especially if you never know, some labs, they have not thinking about accreditation, but if in future they want to go for accreditation, they will be audited, and an auditor can ask, show me your validation data. I want to thank especially the members of IDF Action Group A10, who for years we have been working on this document, and I want to thank in special my co-leader of this action team, Valérie Gaudin, for the nice work together. And thank you all for your attention.
Yeah, thank you, Wim, for your nice presentation.
Thank you.
Questions from the audience? By the moment, seem not. I have two questions, if you allow. The first question is, who verifies the results from the originator lab validation? Is there an external verification of the results and approval or something?
There are bodies who can provide, like AFNOR and AOSC, if the validation was done under their umbrella. You get not only a lab report, but you also get the verification done by these organizations. Yeah.
So then a lab who wants to use these validation data has not to go to every detail of the validation. They can use then the certification by
so
such
these
a
are
bodies.
certification bodies who,
yes.
who will look carefully. There are reviewers checking the data, and so they will certify the original validation study. Yeah.
Yeah, thank you very much. And the second question is why the standard is, let's say, only a technical specification and not a full standard?
Because I see Christian closing his eyes. Was part of the reason, because some people within the group found it was going too far and to oblige everything. So this was a modus vivendi to come to an agreement between the partners in this action team, just to go for technical specification, and that's also in the wording. It's not everything is fully obligatory. Yeah, and so this was a step, a first step. It took already 7-8 years, if I take in mind. So if we really would go for a standard, then I think we had to add many years to this, and we could never come to publication. Not before I'm retiring. Christine is retired
Celia,
now, but
you want
I'm
to
also
add?
retiring. Yeah, this is also, and we decided, let's go for technical specification, and later on we can always upgrade and to make a full standard out of this. Because there was some refusal, some companies and some people along the table, they found this is going much too far. But they were not taking into mind that the first work is only done in one lab in the full world. So, and thinking that this will result that every lab has to go for validation, and there will be more validating instead of testing. And that was the reason, to be honest,
Does these explanations of the presentation of Wim also answer the questions from the beginning, how to validate these kind of test kits? You had this question up front?
Yeah, but his question was also more related for the acceptance criteria in Germany. This is a general document. It's not taking into mind regional or local regulations and so on. He wants to know more about how can I enter in the German market. That was his question.
Thanks for translating, Wim. No, it's also about how the individual dairy actually, document what they're doing.
Yeah.
So if I was auditing a dairy, how would I, evaluate whether the test results was actually happening or not, if there's no documentation requirements in the standards? So partly to the one and partly not.
Okay, next time for the revision.
Okay.
Thank you so much. Other questions, remarks, please? Also from the chat? No one is by the moment.
Okay.
Yeah. Then thank you very much again, Wim.
Thank you.
And welcome, Sylvia. It's a great pleasure for me to introduce you. Thank you very much. Dr. Silvia Orlandini, very well known to all of us in IDF world. She is a biologist with a long experience, of course, in milk analysis, but also in preparation of reference materials for the dairy sector. For 22 years, she was coordinator of the Italian Breeder Association, Laboratorio Standard Latte, AIA LSL. And since 2000, long time, but nevertheless, she's a very active member of ISO IDF committees, different committees. She was a past chair of the Standing Committee on Statistics and Automation, and still chairing the action team on statistics. And since 2015, Silvia is a milk analysis certification executive for ICAR. ICAR is the International Committee for Animal Recording, and consultant for the Milchprüfung Bayern, thank you, in Germany, following the implementation of the ICAR proficiency tests and the improvement of the microscope method to count somatic cells in milk, respectively. Thank you, Silvia, to present us today the somatic cell counting international harmonization by the reference material ECGRC ERMB 0001 and the technical innovations for the future. Welcome, Silvia, and thanks for your presentation.
Thank you very much for this nice introduction. Thank you to the organizing committee to give me this opportunity that really, in preparing this presentation, I had two types of feeling. A small emotion, because I'm getting older, because we have a lot to refer, but especially because what I'm going to refer really is an honor that I'm speaking also on behalf of many colleagues that we work in a wonderful way. And as Dr. Baumgarten just said, nothing can pay such type of collaboration, and really we growing up together in this professional area. So, sorry, the title was likely biased by your introduction, just because on the website I found it, but the concept is exactly the same. So this is a brief outline of my presentation, a short introduction of ICAR, the somatic cell counting parameters. Many of you knows what does it means, but just a short remind. It was the project, the reference system for somatic cell counting, and the tools we have developed in this year. So international committees for animal recording would like to promote the development and improvement of the activities in performance recording and evaluation of farm livestock. And it deals with animal identification, parentage registration. Of course, you can do only if you identify correctly the animals, and the performance recording and evaluation. And it's here that the milk test cover an important rules. So the historical parameters that we considered to evaluate the performance recording of the animals is a fat, protein, lactose, urea, and somatic cell countings. And this is what I'm going to present. As many of you well know, the somatic cell in milk are represented by epithelial cell, a leukocyte, a macrophage, a lymphocyte, and polymorphonuclears, neutrophils. And they rapidly increase when there is an infection in the mammalian gland. So from the blood through the mammalian gland barrier migrates in the mammalian gland to protect the organ. And the somatic cell counting is considered parameters and indicators extremely important and reported in the food legislation, as it was already underlined, is considered in milk payment, and also in the farm management decision and in inbreeding programs. And as you have seen from the previous presentation, millions and millions of tests are done over the globe about these parameters. What it was the problem? So over the years, we realized in the 20s that there was a large bias between the labs. So over the globe, this is an international proficiency testing scheme, we have seen that analyzing the same milk test, the labs have different results, bias plus minus 20, in some cases also plus minus 40. This was not really acceptable in a standardization words, so there was no equivalence. But what to do and how to do? So this was the image, the picture that we had very clear in our mind, and we have to find a solution and to create a dedicated action team in the IDF in collaboration with ICAR. and is called the Reference System for Somatic Cell Counting. This project was presented several times, but I'm very proud to summarize briefly what it was done and the results that we have reached in the last years. So this group, this action team, was composed by 30 members, mainly over the years, and represent 4 countries and 4 continents and 14 countries, but especially what it was important that around the table of this project there were manufacturers, secondary reference material producer, so what we use in the lab to adjust our instrument, non-governmental organization, governmental organization, research center that bring a lot of fresh air and new technology in the project, and especially the routine labs, so the user of the instrument and the labs that produce the analytical data. So it was really nice to collect the different opinions from the different stakeholders involved. The aim of this project, this was one of our picture reported in different countries, was to promote somatic cell counting equivalence and acceptance of the new reference system. What we mean as a new reference system is always we describe it as an architecture where we can bring the information, valid information, and we can together bring something that promotes the equivalence and traceability at the international level. And in doing that, knowing exactly the impact that somatic cell counting has, we invested a lot of energies in communication because we, especially the project leader, Harry van den Berger, Dr. Baumgartner, we were aware that if we didn't prepare the fertile soil to accept the results of this project, we will be not successful. So we presented this project in several countries, and what we use, very valid tools to start the project, what you use the questionnaire. When you send out a questionnaire, you required from the replier a very active reading. So they have to read the questionnaire and they have to reply. And what we received from more than 200 laboratory distributors over the globe was the information about what type of reference material they use, how they use, what type of quality assurance procedure they applied. And please have in mind that all this scenario happened when, at the moment, we have a very valid ISO standard. There is a ISO 13366 Part 2, or IDF 148 Part 2. It's extremely valuable standard, but only the standard was not enough to promote equivalence. So we have to do something more. And also the project have been presented in 20 countries, distributed in all the continents. Also we have done on-site visit to non-governmental organizations, for example, the United States and New Zealand. So any time that we had the opportunity to contact the local stakeholders, we tried to prepare the awareness of this topic. And what it was also important, the newsletters and the communication many times from the group have been translated in the local language. This was really essential, and especially the members of each country, as probably each of you are, have a lot of contacts in your countries, and probably if the message is delivered by you instead from me or another person that I'm not well known in your country, the message is for sure is better received. So this was our communication strategy. Thanks to this collaboration, we started to work in collaboration also with the European community. It's a joint research center, is their central lab in Geel. And in March 2020, thanks to all the expertise that all the members of the group brings on the table, we have realized and we have produced a certified reference material that is available since March 2020. This was really a great result. Again, so we had around the table the secondary reference material producer. Each of them, I would like to say each of us, because previously I was a secondary reference material producer, brings our technique, our experience, the advantage to use such a matrix or such range, and the drawbacks. So we try to merge all the best, and thanks to extremely fruitful and valuable collaboration with Dr. Reinhard Zeleny from the Joint Research Center, we realized that this project is a homogeneous, stable, with a long shelf life, that can be used directly by the labs or can be used by the secondary reference material producer to have an aligned secondary reference material characterized. In the characterization of these certified reference material participated 21 countries and participated with both methods, the reference method that is a microscope method and alternative method that is follow optoelectronic methods. We were in favor also to produce two types of certificate. One certificate was the results, merging the results and the appropriate statistical treatment on only microscope results. And the second certificate was 50% microscope results and 50% fluoroptoelectronic results. In terms of assigned values, we didn't see a big difference. They are very similar, these values, but in terms of associated uncertainty, there is a big difference. And again, here we are with the concept, use the reference material for your purposes. So if I have to train the technicians, for example, on the microscope method, I will use the certificate only, certified, characterized only with the microscope method. If I have to adjust an instrument, could be better to use the second certificate where also the instrument the values from the instrument have been considered. Just the last note of this certified reference material, we have two batches, one low concentration and high concentration. Both are characterized, and what the lab have to do is simply to dilute and to create the range to check the instrument calibration and to calculate mathematically the intermediate values. So this allowed also to check the linearity of the instrument. So in the 2016, ICAR takes the gloves to manage directly the international proficiency testing scheme. And again, the picture was the same, a large variance between the labs. What we noticed and what the laboratories started to become aware. If, for example, I consider this laboratory, at the national level, this laboratory was perfectly aligned with his population, with his group of participants. So no problem, perfect Z-score, perfect performance. Only when he participated in international PT, oops, what's happening? I'm overestimating compared with the other labs. So this was the problem that's starting to be presented. And this was, again, not acceptable. We have to find a solution. We knew what it was the problem. Each color in this graphic represents different secondary reference material. It means that you can see, for example, yellow or blue are grouped in the same area of the graphic. And it means that the different secondary reference material were not traceable each other. And this was the problem. It means that the instrument have been calibrated in different way. So what is we noticed? This is a proficiency testing scheme of ICAR. The filled dot is the last proficiency testing scheme data, so starting from 2016 to September 2009. The clear blue dot is the distribution in 2019. It's still large, so you can see with the laser, you can see this lab is really far, but as I told you, in March 2020, the certified reference material starting to be distributed on the market, and you can see that in September 2020, only 6 months later, without to impose anything, just because the golden standard was on the market, the population started to become closer to the center distribution. And if we go ahead, there was a further improvement in 2021, and this is September 2021. The red dots, really, it was really, for me, a great surprise. I would never expected in less than 1 year to have such an improvement. So really, these results, the use of the golden, or the possibility to use the golden standard, put the labs in a position to start to create a traceability, start to build up an international traceability. Of course, also the precision of this distribution increased a lot, improved. The red line is the standard deviation of reproducibility reported on the ISO standard. And you can see that in the NICAR proficiency test, we never fitted before. Now, finally, in the last 2 PT, we are below. Just a note, how the limits of the ISO standard have been established in the past. When we revise this ISO standard, we considered the different proficiency tests around the globe. And again, each proficiency test mainly was organized around only one secondary reference material. And of course, the precision was better than an international proficiency testing scheme. So now, finally, demonstrating that we are below this line, it means that really we are aligned each other. So this is the perfect example of how the statistics became real in the real life. So the reference system, and again, the somatic cell, I would like to underline that is just an example, and we consider this parameter because we knew the problem and we tried to solve. And it's really a lively example on what the IDF ICAR strategy would like to promote. We know that in theories, so as it was presented by the president at the beginning of this session, we would like to extend the consensus platform with a new initiative for the benefit of the entire dairy sector. We would like to enhance the collaboration and to share experience in analytical fields. And at the end, we would like to have international traceability and equivalence again. So the work is not finished. We would like to continue. We are extremely happy and satisfied, but we need to do more and to continue always with the same philosophy. So we have several initiatives in the ICAR and the IDF. In food, nutrition, and security is a EU project, and we have started some method to consider some method implementation. So what it was done in ICAR IDF, we have published a bulletin. We really thought that was extremely important to provide a guidance on how to use this certified reference material, not in terms on how to prepare and to dilute, but in terms on how to evaluate the data and how to adjust the calibration of the instrument. This bulletin is not an ISO standard. It would like to be just a guidance, would like to be only a support for the lab, and also we provided an Excel file where the lab can simply input the data and to evaluate if it is the case to adjust the calibration of the instrument or not. So it can do really or virtually, just mathematically, to have an idea what can happen. And again, it's an example. But it seems that from the experience, from the feedback that I received from some lab is easy to use, and the results of the international ICAR PT seems that it works. What we are doing, also we are asking to the participant information of the type of secondary reference material they are using. And we are now going to produce another questionnaire in SO9 because we would like to see how they are building up the traceability. Because again, we have the guideline, but we would like to be sure that they apply these or other methods that are aligned each other. So again, the questionnaire for us is extremely important tools. And the last point is that this week we're starting to revise the ISO standard part 2, this fluoro-optoelectronic method. There are several reasons, some very simple as the technical. So for example, the displaying on disk should be not anymore reported in the ISO standard because it's not used anymore in the field. But what we would like to introduce is the concept to make aware the labs and the international community that there is a certified reference material available. In practical aspect, this means that if a lab is accredited, when it will be audited, the auditor will read that a certified reference material is available, and it means that the lab has to demonstrate that or use directly the certified reference material or it using a secondary reference material that is traceable with the certified reference material. So this will be a further step to promote equivalence. Another aspect was that this EU project supported IOS, that is my company, Analytical Equivalence. It's not the case that I choose this name. And Chemstat is Werner Lengible, is my colleague. We worked together starting from their publication of Thomas Berger and Werner, where they presented a probabilistic approach to qualify the labs in a proficiency testing scheme, but also to qualify the proficiency testing scheme itself. So applying this model, we can assign a quality index to the lab and a quality index to the PT scheme. To simplify the statistical approach, the common things is that, first of all the data have to be treated according to 5725, that is a standard, where it is described how to do the statistical data, to treat the statistical data, and also that all the data are compared with the precision published in the ISO standard for the somatic cell counting. So this example was done and optimized for the somatic cell counting. One time that we had this publication, thanks to this EU project, we developed a software, dedicated software, that can, if the data are presented in the right way, can process rapidly thousands and thousands of data. The test that we have done to prove the software is to collect the data to the from five PT providers that kindly have trust in this project, trusted in this project, and provided us the data that have been completely anonymized. So the results of this project that is going to be produced will be completely anonymous. But the result is this. So the histogram is very well known, many of you, but this quality index is ranked from 0 to 1. Closer is to 1, the lab, better is its performance. And the same to qualify the PT scheme. Closer is to 1 and better is the performance. So you can see, back here. You can see here for example, these are the PT scheme close to 0 because they have a large variance, so very far from the reproducibility published in the ISO standard. Here we have two PT providers with a very good index. What we have done also to have opened the possibility to this software to include some premium. And this premium as, for example, the use of the certified reference material. So if a laboratory report that his instrument performed the PT and the instrument was adjusted according to the golden standard, he receives a premium, and so he will have a closer results to 1. And the same for the PT. If the proficiency testing scheme assigned value is traceable with the gold standard, the qualification of this PT scheme will be closer to 1. What is good that this software has been bought by Enea, is an Italian research center that is the coordinator of Metrofood, that is a big and huge European project. And they would like to extend and to apply this probabilistic approach to other matrices in the food. Because as you well know, the traceability in the agro-food sector is really underdeveloped. So if we haven't a gold standard as we have obtained now only for the somatic cell count, we need other tools to compare each other. Next. Other aspect we considered to start to implement the microscope method. Does it make sense that we are continuous to use the method that was starting to be applied at the beginning of the past century? So nowadays we have the technology to do that. In my opinion, we cannot suggest to apply too much sophisticated technology because we have also to deal with raw milk and with the routine methods, so we have to find a compromise. So this project, the microscopy image analysis in milk, is called Miami project. It was the project that I have the honor to coordinate, thanks to the MPR and QLEAP that provided the microscope and the digital camera associated with dedicated software, and to the support from some partners, from some manufacturer, Page and Pedersen, Lactoscon, and Foss. So what we have focused our attention is to the historical dye solution that was methylene blue and dry smear. To dry the smear under the microscope, we have tried to find other dye solution and to simplify the procedure of sample preparation using, for example, directly liquid milk. So the main purposes of the Miami, of course, was to implement the reference method to create image, digital image that can really be an objective evidence of what we are reading. So the technician, when they put his eyes under the microscope, on the microscope, is starting to count, and he obtain a number, but nobody knows what really he counted. So with the digital image, we can see one by one. You can see cross one by one the check, and you can store this image. And can you imagine this, how much is valuable when we characterize a certified reference material as we have nowadays, or how much is important to compare these results with another batch. So this will be really a valuable tool. So what we have tested and we have proven that is possible to apply is a liquid milk, to use a liquid milk in a disposable counting chamber. Ethidium bromide as well works very well also with liquid milk, but in a more concentrated solution. I have indicated in this one with the 2. And the YoPro one also is a new dye solution with a good fluorescence that spot out very well the cell under the microscope. This is the pictures. I would like just to call your attention on the fact that, okay, we worked with excellent technicians, with excellent repeatability, and also with, I can see the laser, what is it? You see there? Here I can see also there. Okay, the dry smear, okay, the dry smear has a very good repeatability below the ISO standard part 2. So this is the precision of microscope method. Here we are much below. And also with the new techniques, this means automatic manual reading. It means that we capture the image and manually we counted on the digital image one by one the cell. And also in that case, we obtain a very good repeatability on dry smear and liquid milk with ethidium bromide and with YoPro 1. And also the difference from certified reference material, what does it mean? So we analyze this sample, single cows, secondary reference material, bulk milk. We analyze it with a fluoro-optoelectronic instrument where the calibration was adjusted with the golden standard. We assigned the values, and we compared these values with the results under the microscope. And also in this case, we are below 10% with all the solution tested. And for us, this was really a great results. And in any case, was really, I mean, it's very close on what we are using with the fluoro-optoelectronic instrument that is an extremely precise method. This is the slide of the last test that we have done. We call it the phase 3. When we extended our comparison to the fluoro-optoelectronic technique, I'm going to present to you some technique briefly in the next slide. In the microscope, we tested the comparison of the new technique with the historical technique, is methylene blue dry smear, and the comparison with the golden standard was extremely good. And this part is still under development, but it seems to show very good results. This is the second technicians that didn't prepare the sample, simply have taken the digital image and is starting to counting. So despite the COVID, despite the pandemic, we really went on the project and we produced results thanks to the technology and sharing the digital image, and the results of the comparison also is very good. So we can imagine the future to share instead the samples with all the difficult to share image. So this was a very good results. Just an input that I didn't remind, during the week these results will be presented in the statistics and automation, and it's our intention to try to propose the revision of the ISO standard part 1, not in terms of revolution, but just to introduce this innovation. I would like to be more than satisfied as an annex, for example, because it can be the way to starting to prepare the labs to apply these new methods, and in the future to promote a new collaborative study and new precision data. So this is a proposal that we will do during this week. Another technical innovation is a project that again will be presented during this analytical week. Is a Antoinette project, is developed at Agroscope, and is the development of a new flow cytometric technique. And what is great is that at the same time this technique can allow to do total somatic cell counting and differential somatic cell counting with flow cytometry without to prepare the sample. You know that there are a lot of bibliography when we can apply the flow cytometry with the use of antibody, but the sample have to be treated, have to be centrifuged, and the cell have to be isolated. We have to treat the matrix, so it's completely different approach. Thanks to this great project, the matrix effect can be deleted, the noise at the background, and the cell can be, the counting can be obtained. The results are quite comparable with the total somatic cell counting flow cytometer method, and also is possible to differentiate the different population. So it will be better explained by the project leader during this week. So some short conclusion. My experience in this project was really that the communication and consensus are the key words to work in a wonderful way in this action team. I have to say that I was an action team leader of other projects, protein revision, freezing point, Rose Gottlieb, without to take, I mean, also these were very important projects, but they were more technical. The reference system for somatic cell counting, it was much more. It was not only technical, it was communication, it was good politics in terms of consensus, but it was really, it was the proof how much is important to prepare the audience before to deliver the final results. So my last big thank to all the colleagues that participated in these years to this project, and thank you for the attention.
Thank you very much for this presentation. Any questions?
I would like to know what about the next batch of certified reference material, if there is any budget for this, and until now, do you think we will be able to use the current batch?
There is a big store still, so we are not in a situation that we have to start to produce a new one, but we already make sensible the European community about this problem, so how it will be managed in the future, so they are aware of it, and so they have an alert level. Then when the batch will be a level that they have to start a new characterization, so they are aware of this. So I'm quite comfortable for it. In any case, all the procedure has been described. So we have all the elements to follow. For sure, we have to monitor over the time.
Okay. Any other question? No?
Then a big thanks to Sylvia also for your last words to encourage really to be active as in the action teams, and for your positive outcomes. Thanks a lot for this, Sylvia. Thank you.
So this leads us to the last lecture held by two speakers. First, Karin Grehenbühl is currently operational chemical food safety manager for Nestle Infant Nutrition in Switzerland. And she joined Nestle in 1999, where she held several positions with focus on food process, food flavor, food bioactivities analytics, and quality safety. And at IDF, she chairs SCRCC, Standing Committee for Residues and Chemical Contaminants. And besides, she contributes to Codex and Zen transversal working groups on food chemical contaminants, and chairs the SNE, the Specialized Nutrition Europe Working Group on Food Safety. Thank you very much for your to be here. And?
And it will be a shared presentation, as I understood, together with Dr. Harry van den Bayard. Harry studied food science and technology at Wageningen Agricultural University, and present he is a manager for innovation and business development with QLIB in the Netherlands, a provider of QA services in the global dairy sector. Yeah, Harry is very well known as a very active member of the international harmonization of methods in analysis and sampling of milk and milk products. He is the past chair of the IDF Method Standard Steering Group, MSSG, the past chair of ISO TC 34 SC5, and of CEN TC 302, and all of the methodology of analysis and sampling of milk and milk products. And he is still an active member of the Standing Committee on Residues and Chemical Contaminants, and in 2018 he was appointed as a board member in IDF. Welcome also to Harry. But you will start, Karin. Thank you.
So, thanks a lot for the invitation, for the kind introduction, and we decided to split this with two voices, because we are trying to take you through a number of different aspects related to chlorate and perchlorate, and we will touch on regulatory aspects, food safety aspects, then also maybe some words about the origin, the root causes, and the ways to mitigate. And in all these aspects, we will try to show where the IDF can contribute with guidelines and harmonization. And this will bring us to also the analytical part, where harmonization, of course, is very important, especially when it comes to enforcement of limits. So chlorate and perchlorate, they are often mentioned jointly. But there are some differences and some similarities, so we will try to show this over this presentation. The molecules are shown here in this first slide. They are very small molecules, charged anions, water-soluble, very similar in structure, only differing by the level of oxidation. But in the dairy chain, actually, the origin is quite different. So chlorate occurs mainly, and we will go more into the details in the coming slides, but it's mainly by contact with chlorinated water, so it's linked to the microbiological safety mainly. And other, not only direct chlorination, but also chlorination by chlorine-based sanitizing agents. So other chlorine-based sanitizers also release chlorate. Perchlorate, on the other hand, is persistent in the soil and is mostly carried over from feed to the milk through the grazing or the direct feeding. But what they share is their mode of action in vivo, so in the body. They both act via the same mechanism of action, and they interact with the iodine absorption in the thyroid gland, which may then have an impact on some aspects such as growth and development. But perchlorate is 10 times more potent, so it's the same mode of action, but one of the residues is more potent. And in terms of regulatory approaches, we have seen a lot of different approaches worldwide. Some regions or countries have set strict regulatory limits, and other countries have not set limits at all. And it's of course linked also to the responsible management of the microbiological safety, this balance between what can be allowed or what should be regulated. So this first slide is the overview, and now we'll go a bit more into the details. So what is the food safety concern? We have a number of tolerable daily intakes and other health-based guidance values defined already by global or local food safety authorities, mainly by the GECFA, by EFSA. These are intended to define what is acceptable in a regular diet on a daily basis. And so helps also setting possible regulatory limits, especially for sensitive population. And it gives also some guidance to which level these substances should be mitigated or reduced. So historically, we had a number of food safety opinions published. They are cited here. But there are some recent developments also. So maybe what I can say here is that this subject is still a bit open to controversy. Also, there are some very conservative approaches that have led to very strict conclusions that are maybe a bit more attenuated in recent publications. Especially there are publications 21 on benchmark dose modeling that lead to less strict conclusions with regards to what is acceptable in terms of daily intake. And we also have a review paper by ANSES just very recently. And also now the question is open at European level whether the 2015 scientific opinion should be revised also based on these new findings that are in public literature. So experts still, let's say, discussing what is relevant in terms of setting health-based guidance values. And this applies to both, as I said, perchlorate and chlorate have the same mechanism of action in vivo. Then the regulatory aspect. So here you see a number of organizations have set guidance, especially for water. Drinking water is regulated and has also given a number of guidelines, such as the WHO Drinking Water Quality Guideline, which sets a level for the potable water at 0.7 milligram per liter, so quite high. We have similar levels set in the Chinese regulation, and in the European Water Directive, slightly lower level here at 0.25. So also here, I would say the international experts and organizations not fully aligned. Then in foods, we have this strange situation, and I would say sometimes conflicting regulation in Europe that chlorate is considered as a pesticide, because in the past, more than 15 years ago, it was used as a plant protection product, and especially also as a desiccant on cereals in the final stages of the before harvest, because it dries out the grains, and in this context is still used in certain areas in the world. But in Europe it's been banned for, I think, over 15 years now. But we have this regulation that says that every pesticide that has been authorized or used in the past as a pesticide will still be regulated as a pesticide, and that's why this umbrella regulation, the 396/2005, so it's a hyperlink. When you get the presentation, you can click on it if you're interested. It's actually setting the generic limit at 0.01 milligram per kilo, as consumed especially for the young children, to protect them against the influence of pesticides, which in this case also covers chlorate, and that's where the strict limit on chlorate comes from in Europe. And in other areas of the world, we don't see this strict regulation on milk or on dairy finished product. For example, we see that in New Zealand there is a publication recently setting the action limit much higher at 0.1, and in infant formula at 0.4, which is then in disagreement, I would say, with the approach that is used in Europe, for example. And this has made, I would say, the management of chlorate residual levels quite a challenge in the last 7, 8, 10 years almost, I can say. And the reason is that the entry points are numerous into the chain of the finished dairy products. And here I show some steps where you can enter the product. So already at the farm, due to the use of chlorinated water for the equipment cleaning, and then at different steps where the contact materials need to be cleaned, containers or trucks, all the transport facilities. Then in any factory where there is a transformation step, a concentration step especially, and even more so when there is water used as an ingredient that is then evaporated, leading to another concentration, because the municipal water is also in many places chlorinated. And finally, there is also, and here I show the example of an infant formula, there is also a contribution at reconstitution in the households when the powder is then diluted with tap water, which is recommended in most geographies. It's safe to use tap water for the reconstitution of infant formula, and that's how it should be. But what we can say along this entire chain, there is no source that comes from pesticide treatment. It's really always the water that contributes to the residual level at the different steps. And as a consequence, there are a few papers showing some distribution and occurrence of chlorate in dairy products. The variability, the levels that are found are very different depending on the countries and the geographies where the measurement is done or the studies. And you can see here from this 2016 paper from Ketlitz and colleagues that when samples are sourced from the southern regions of Europe, we have higher levels than when they are sourced and measured from the northern European countries, because the chlorination of the water is also very different, and that's linked to temperature, climate, and the management of the microbiological safety. And when we go out of Europe, for example, Japan or USA, you can see that the levels are again in a completely different range, the values that are found in the water. And as a consequence, in the milk products, depending on where they are produced and also where they are transformed into powders, for example, or into advanced dairy products such as whey protein concentrates, you will also find quite a variability of the levels. And you can see that this goes from not quantifiable below the 10 microgram per kilo up to several milligram per kilo, which are real values. Okay, they were found a few years ago when the discussion started around this topic, but they were really measured. And still today there is a big variation between different regions in the world with regards to chlorate residual level in dairy powders, for example, simple skim milk powder or simple whey protein concentrate. And this depends, of course, on the level in the fresh milk, and then also on the process that is applied, the processing aids, and the chlorination practices that are used locally to manage the ingredient water. Now, that's just an example to show how it reflects on the finished product that is dear to my heart, is the infant formula. So you can see that of course, here I'm showing that pesticides is not a root cause in that finished product. It's really the hygiene aspect and the management of the water that leads to the values that are found. And each of the ingredients that are added into infant formula, for example, contributes to a certain level because the ingredient itself is exposed to chlorate at some point in its manufacturing process. So that's also the conclusions that all the manufacturers ended up finding when the whole discussion started in the 2015. Between 2010 and 2015, there was a lot of discussion around this topic. And the initial values that were found then very high were gradually reduced by acting on all these ingredients, including the dairy ingredient, by good management of the chlorination steps. And that's where we will have the need also to have harmonized analytical techniques to be able to decide fact-based between the manufacturer of the ingredients and the buyer of these ingredients that will use them in their processes to make sure that the analytics is solid and reliable and can be then used to establish purchasing limits that will in the end ensure a compliant finished product. So, but that's the level of complexity that can occur in a finished product when there are several ingredients plus the factory ingredient water that come together to add up to give a finished product value in the product that goes on shelf finally. And so the manufacturers have engaged with the Commission in Europe on a gradual mitigation plan. It's called the roadmap, where every year the values are reduced, and with the help of all the ingredient suppliers, including the dairy supply chain. And you can see that all the infant and young child products have been now brought to lower levels, which has taken a couple of years, but is now close to the target, and was really not an easy task from the beginning. So how was it achieved? So it's really important to act upstream here, both at factory and at farm, and to manage properly the chlorination of the water. So it's well known by now that hypochlorite upon storage will generate chlorate, as it's the one main side product, and this is dependent on a number of parameters, including the time, the temperature, and the sunlight exposure. So with these parameters well controlled, there is a way to reduce already the formation of chlorate in the hypochlorite that is used for the chlorination to manage the microbiological safety of the water. So that's the first point that can be managed. Then the other aspect that sometimes is needed to have low-level chlorate in the water is in some areas where the chlorination of municipal water is really high, there may be a need to dechlorinate the water first with reverse osmosis, and then to add a controlled level of chlorate with an automatic electrochlorination type of chlorination agent. That's something that has also brought very good results. And in composite foods, it's important to look at the contribution from all the ingredients and the processing aids. It is known that caustic can contribute chlorate levels. Also, some salts are significant contributors. So all these aspects need to be managed simultaneously. And that's also what we are trying to summarize in the technical guidance papers we are developing with the IDF, all these good practices and ways to mitigate the contamination while maintaining, of course, a safe, microbiologically safe environment, and balancing the two aspects to be optimal on both sides. So that was about chlorate. Now on perchlorate, the regulatory aspects are slightly different. We also have guidelines in drinking water, but from WHO and from the US, but not from China or Europe. And we have a strict regulation in Europe on perchlorate. It's regulated as a contaminant, and also some limits set in specific finished product when they are intended for infant and young children. Currently, and that's an anecdote, but it's important for people who are working with this on a daily basis. Currently, we have contradiction at European level between the regulation that is set on infant cereals the regulatory documents that are available from the Commission are not consistent, and this has been brought to the attention of the Commission and should be corrected. So when you go to the intranet site, you will get two contradictory information depending on where you click on the hyperlink, which is a bit of a concern, also leads to some disputes between some key stakeholders in the manufacturing chain. So you can find this information here, and we hope this will be settled soon. And the Commission has taken note and will fix this, hopefully as soon as possible. And then, with regards to the entry points for perchlorate, as I mentioned, it's naturally present in the soil or can occur from environmental contamination, meaning from the specific local contamination, for example, from rocket fuels, propellants, explosive fireworks. So this is really generally very local. But more frequently, we see contamination from nitrate fertilizers who can be rich in perchlorate. Also from some processing cleaning aids. And this will add up then into the feed of the cows and will carry over into the milk. That's the main root cause for the occurrence of perchlorate in the dairy chain. So now I think with this I would hand over to Harry for the analytical aspects, because it's very important then the contribution that we have from the IDF to make sure that all the standards are in place to determine.
Thank you, Karin. 5:45 almost. I was thinking about the words of Christian, and I'm going to paraphrase a little bit. One of your last words of wisdom in your speech was, what do you want to know? That's the question that we have to ask. But I wonder at this moment, do you still want to know? I hope you're still awake. Anyway, I will take you through some other aspects of chlorate and perchlorate in dairy matrices, and I will focus indeed on analytical aspects, but I will also talk a little bit about strategies to mitigate, indeed also through testing, and furthermore on an initiative in AOC, but also in IDF on chlorate, perchlorate, but also on emerging contaminant risks in more general. I will try to be fast, don't worry. I will start with the same wording as Karin. It's small polar molecules, and that puts challenges in analytics. And in literature, one can find methods on different principles, and these are listed here on the screen. And these were mainly developed with water as the target matrix. And of course, these techniques are capable of measuring at the levels relevant for water. The limits are higher as in food matrices, especially infant formula, but not only water, also in urine and serum. But to check compliance with its strict limits for certain foods and also the raw materials for these, we have to find other ways to get accurate and precise results at lower levels, and therefore at the moment LC-MS is the method of choice. And I'm looking here at all the test kit providers. Is anybody working on a test for chlorate at the moment already? You see options? Nobody wants to answer, of course, but at least I hope it makes you think about possible solution, because I think there's an opportunity for you here. The method of choice, yes, LC-MS, and we developed one at QLUB, started that a few years ago, and I want to share some of our experiences with the development of that method. I know it's also underway or has been done in other laboratories, but the biggest challenge in developing a sensitive method in dairy matrices or food matrices in general is the avoidance of interferences. And there's many in milk. Think about fat, protein, lactose. These put a burden, but also another possible interference is with contamination coming from filters, solid phase contact materials, or laboratory equipment in more general, laboratory equipment that has been rinsed with water. That may interfere. Fat can be removed from milk, we know that. You can centrifuge, or you can go to more costly solid phase extraction steps. You can use cold ethanol or acetone to precipitate milk proteins in the solution or from the solution and apply centrifugation to remove them. And then you can isolate the supernatant and dry and dissolve the pellet and then pull them over a C18 column. That's more or less the principle of the method that we apply, and which is not really an original method, because the basis also for our method and many other related methods is in the work of the EU reference lab for single residues methods in Stuttgart. That's the town I passed by on my way here. Not only that basis, which we simply took from the internet, but also I should here mention the valuable tips from Martin Denneher from TGASC. And that's why you should attend Analytical Weeks, because he presented that in the Analytical Week in 2018 in Dublin. So that's where it comes from, that QLab method. To give you some idea of the performance of the method, we have it operational since early 2020, and the performance is here. Quantification level about the same as presented in 2018 by Martin. And you find here an LOQ, a limit of quantification. That's where that's the minimum level from which we dare to report quantitative data. That's at the 2 ppb level, so that's a considerable margin towards the lowest limits. And you see also listed here limits of quantification for dairy matrices, and these are listed under the table for both glutarate and perglutarate. And that means that with this method we can indeed check compliance at different levels if we want that. Recovery is good. Repeatability, measurement, uncertainty are, I think, quite acceptable, and we will see that later when we compare against the method performance requirement of AOEC. The method is submitted for ISO 17025 accreditation. That was a short advertisement. Take it as you want it. Anyway, there are standardization efforts underway because, as I indicated, there's at the moment many laboratories investing in this method as a single method for glutarate, perglutarate, but also as part of multi-substance testing. And it's also the reason that AOEC picked it up, and they have defined or developed, I should say, Steve, a list of standard method performance requirements for these type of methods, and then focusing on the target matrices in baby foods and infant formula. And there, in the table on the screen, you find the demand with regard to the LOQ for different matrices, also the demand on recovery, and the demands for the relative standard deviation of repeatability and reproducibility. And if you have still the former table in mind, remember I listed the repeatability as 2.8 times this standard deviation. This work in AOEC is also picked up in IDF ISO, and that's a discussion point for tomorrow in the meeting of analytical methods for additives and contaminants, CAMAC, whether and to what extent IDF should hook up to this AOEC initiative in developing or co-developing with AOEC a method under the IDF ISO umbrella. What I also should mention here is the becoming available of secondary reference materials that is coming through NIST. NIST has got 2 reference materials available where at least there's a certified value for glutarate available now. It's all the materials. I think the last one is younger than the first one, but both come with values for glutarate now. So that's already available for those who are aiming to test on glutarate and perglutarate. A slide on testing strategies. Yes, as Karen pointed out, part of a successful mitigation of the issue is in root cause investigation, and that's what I think many around us and maybe we ourselves are aiming for. And in the dairy value chain, we are dealing with raw milk, of course, as a raw material, and that's a potential source for chlorate, but certainly also for perchlorate, which comes into the chain through feed and water, also from the environment. But it also means that it can come from many potential sources, individual farms. And those individual farms may show a large variation in the burden put on the herd bulk milk that flows into processing. So it's feeding practices, it's sanitation practices on individual farms that determine the load on the milk for processing. And that also puts the challenge to the processors with that input already to meet with those regulatory limits, especially for infant formula. LCMS, as indicated, is an expensive method relatively, but luckily we can get to low levels, and that means that we have a margin there to commingle samples. So if we have the cost of test, it goes quite somewhat beyond €100, then you try to save cost of analysis through commingling samples and then find from the result in the commingled samples whether there's any further action needed in the underlying sample. So a two-step process. What is relevant in there is what is the gap you have between the limit of quantification and the level you want to signal in the individual sample. So what level you want to detect in individual farms, and we can imagine an approach. Karen mentioned it. If you gradually lower your limit, you can first try to pick out the highest contaminating farms, and then gradually go to lower levels and go to a situation where you have a continuous improvement. What we also have to take into account when commingling samples is that we are dealing here with background levels of chlorate and perchlorate. That's more or less a level here. You find almost in every sample you find something, especially when you go to low detection levels. And that means that you have to also take that into account when making your calculations for commingled samples. Yeah, a formula at this time of the day, that's not good. But the slides will be shared, and what I did is simply gave you this formula with the full explanation and also the conversion of the formula in the calculation of the numbers samples you can put together, and the tables serve to illustrate how that works out. And I'm not going to explain it in full because it's too complicated now, but anyway, if you take 20 minutes tomorrow morning early, not after the reception, but tomorrow morning early, you go over it, you will understand what is showed here. So it comes down to when you're less keen to detect low levels, that means a higher signaling limit, you can work with a higher action limit for your commingled sample. That's the first table. And that's already a lot of words, but that means that there is more room, so to say. More background contamination urges you to do the same. And when commingling more samples, that's the vertical one, you must act already on a lower action limit when you want to detect an individual non-compliance with the signaling limit. So that's, I see Steve dropping out now. Okay, then I won't explain any further, but please look at the formula, try to understand it, and you can find the examples in the table, and then you will also understand the principles behind making use of commingled samples. That not only works for chlorate and perchlorate, but also works for other residues and contaminants if you have to run a residue monitoring or survey program. It's a strategy currently that we apply in the Netherlands to learn about the root causes, because we are in the learning curve. Chlorate we know, but especially perchlorate puzzles us. Where does it come from? Nitrate fertilization, but we see a tendency, there's differences in regions, and we do not have a clue yet. We need more data, and that's what we try to collect this way in a cost-effective manner. So that learning curve will bring interesting new information and hopefully also further options for mitigation. So that brings me to a summary, summarizing the parts of Karen and myself, and just stay seated after the summary because then I have a few slides on the IDF work. Glorate and perglorate, yeah, you can read for yourself. I don't have to read it for you, so simply look this through this bullet and you will find hopefully what you have already picked up from the presentation thus far. So that's what's listed here. So it's a combination of what is the mode of action, what are the entry pathways, these are different regulatory approaches, costly methods thus far, and think about the proper strategy to detect root causes. Okay, this work on glorate. Karen has been the project leader on this topic as part of the work of the IDF action team on emerging chemical contaminant risk from farm to processing. And that's why I wanted Karen to present her work in this together with me. She has done an extensive effort there, and hopefully that will soon come out as a deliverable. We talk about that today or tomorrow further. The purpose of that action team is to build topical guidance on emerging contaminants, not only on glorate and perglorate. There's more, and I will have examples in a minute. But there's a number of IDF standing committees involved. It's cross-sectional activities. Residues and chemical contaminants, that's the standing committee leading the effort. But there's input also from farm management. Think about a lot of those residues and contaminants coming in through the farm. Think about microbiological hygiene. Clear, glorate, perglorate, you want to avoid it, but at the same time you have to make sure that your products are safe. Additives and contaminants, analytics, but also dairy science and technology. Maybe not so much here, although in disinfection processes in dairy technology, that these are related. But if we come to other topics, like here listed, there is topics amongst these where dairy science and technology is most relevant. Here you find different categories, different colors. I'll try to explain a little bit. The categories are clear. I don't have to explain that one. These were the ones that were identified from a questionnaire amongst IDF national committees, also asking them for a top 10, as simple as that. And from these, the bold topics arose as most urgent. And even the 10 that were highlighted, that was too much to take on board immediately altogether. So we decided to pick one as a start, that was the glorate. We soon after also concluded it's worthwhile to go further in other disinfectants, detergents, and also there is work underway on deliverables. And more recently, we have done some work on microplastics, which is now brought under the attention of the SPCC. And we hope they can give us some guidance how to proceed with that topic. But you see there's still a number of bold topics not progressed thus far. Work is still to be conducted. And from the discussion we've had in the SCRCC and also in the SCAMAC, also tomorrow we will have a roundtable. What is at the moment a priority or of relevance in your country? We update this list time by time. It appears that also endocrine disruptors as well as PFAS are in the center of attention in many countries. So that may be next topics, but it all relies on availability of experts. So this is also a call. If there's people in the room interested in those topics, please contact me. I will be around till Thursday night about, and then we can see to get you hooked up to a topic. And I know there's people in the room who have already hooked up, and I'm happy to see them here, but we definitely need more experts on those topics, making sure that on each of the topics we can, as IDF, deliver to our stakeholders in the form of technical papers, more extensive, but also short fact sheets, in order for the less I cannot say less interested, but that's people who take a more sniff of the project, at least that they have a brief understanding, and that's what we have fact sheets for. And that's also part of the aimed at deliverables from this action team. So most welcome, and I think that's it. That's a more nice ending, I think. You're most welcome to join the effort. Thank you.
Yeah, Karin and Harry, thank you very much to both of you for the insights on the detection of chloride and perchlorate in milk as the joint activities to cope with an emerging concern. Maybe there are still some questions. Marcel?
Yes, thank you. Yes, you mentioned in your presentation that NIST is developing reference material for chlorate and perchlorate, if I understand it correctly. Yeah, they have it available. But since it's also an important topic for Europe, do you know of any initiatives of JRC in Gale to also develop these reference materials?
I don't know, and Karen is nodding no, so...
No, there is none now.
Okay, okay, that's clear, thanks.
Any other questions? Maybe from the chat? No? Then thanks again to both of you, and you come back later also to the desk, please. Yes, the present, thanks. Yes. Sure. Ja, Ja, danke. Dankeschön, Max. Thank you. With this, I would like to hand over to Ludwig Bürger from IDF Germany, Verband der Milchwirtschaft. Thank you.
And that's my job, to thank you. And to give a small applause to Ms. Gerten and Mr. Mowarek. Yeah. Many thanks also for you. We have some presents I will give it later to you. We will have a short break, 15 minutes, and then we will have a sponsor session from our platinum sponsor Bentley. It's about latest innovation, analytical and automation solutions for the dairy industry to keep enhancing milk quality, safety and value. So grab a coffee, grab water, come back in 15 minutes. The reception later this evening will start at 7 o'clock, maybe a few minutes later, but please join us outside in the exhibition area. Many thanks again to you, many thanks to the visitors today. See you later.